Motion size.
Prosociality, the preference for outcomes that produce benefits for other individuals, is ubiquitous in humans. Such behaviors are not just restricted to mankind, but can be found throughout the animal realm, from social choice in our close primate relatives to the eusocial communities in insects. Here, we used rats as a model to investigate the evolution and neural substrates of social behavior. We introduced a rodent analog of the prosocial choice task, a simple and standardized behavioral experimental paradigm adapted from a well-established task in primates to probe prosocial choice behavior. Experiments were conducted in a custom-made double T maze with the maze's main compartments facing each other. Each T maze consisted of a starting box connected to two decision chambers by two independently operated doors, each leading to a choice compartment. In this task, we trained an actor and a partner rat, where actor decided between two alternatives only differing in the reward delivered to the partner. The own reward choice resulted to the delivery of 3 sucrose pellets only accessible to the actor, whereas the both reward choice produced an additional 3 pellets for the partner. Thus, the partner's food access was contingent upon the actor's choices. All actors underwent both a partner condition where they were paired with a real rat partner, as well as a toy condition where an inanimate rat toy puppet was used instead, which served as a control for the potential non-social motivational mechanisms. We conjectured that if a conspecific's access to food carries reinforcing value for actor rats, they should develop a preference for the both reward alternative in the partner, but not the toy condition. Using a first batch of 16 animals tested for 10 sessions per condition, we first studied at the group level rats' preferences for both rewards compartments and whether their preferences differed between conditions. The proportion of both reward choices was significantly above chance in the partner condition and significantly below chance in the toy condition. Moreover, there was a significantly higher proportion of both reward choices in the partner condition compared to the toy condition. These results suggest that both reward preferences were dependent on social components of the task. To investigate whether the individual choice allocation pattern was stable over time, we tested a second batch of 32 rats for 7 sessions per condition, and then retested them in a repetition phase of 3 sessions per condition. The percentage of both reward choices was significantly higher than chance for both pre- and post-repetition in the partner condition, but not in the toy condition. Moreover, we found a significantly higher percentage of both reward choices in the partner compared to the toy condition in both pre- and post-repetition phases. The percentage of both reward choices did not significantly differ between pre- and post-repetition in either condition. These results suggest that rats' social context-dependent preferences for the both reward alternative remained stable after a repetition manipulation. Preference for the both reward compartment greatly varied across rats. Some rats showed substantially higher preference for the both reward alternative in the partner condition compared to the toy condition, whereas others neither developed a preference for the both reward alternative nor showed a condition-dependent choice pattern. To determine the extent to which rats differed in their both reward preferences, we calculated a social bias score for each rat reflecting the percent change in both reward choices in the partner condition relative to the both reward choices in the toy condition. A positive social bias score can be interpreted as added positive social value placed on the partner's access to reward, and a negative social bias score can likewise be interpreted as negative social value placed on the partner's access to reward. To better characterize the differences in mutual reward preferences between rats, we compared each rat's social bias score to a distribution of social bias scores computed with shuffled data. This analysis revealed that 60% of the animals had social bias scores above the shuffled distribution, and thus could be classified as prosocial, whereas the remaining rats were categorized as non-prosocial. Body mass, correlated with group hierarchy in rats, has been shown to increase helping behavior towards lighter animals. Therefore, we asked whether the individual differences in social bias scores could be explained by the partner's individual weights. To this end, we correlated the social bias scores with the weight of the partners. We found a negative correlation between normalized partner weight and social bias scores, suggesting that actors had a higher propensity to choose the both reward alternative when paired with lighter partners.